So we oftentimes want to find mutants so that we can run different tests on them. But mutants are rare and they are oftentimes very hard to isolate. So there are two basic approaches we use to isolate mutants. The first is super simple. It's called direct selection. So we select for that mutant to grow. An example of direct selection, here we have a culture that contains some type of bacteria. Some of those bacteria are resistant to streptomycin, while others aren't. So if we grow that onto a medium that doesn't have streptomycin, we're going to get lots of different growth, right? But if we grow it on a medium containing streptomycin, we're only going to get the cells that are streptomycin resistant growing in general. And so this would be your streptomycin resistant cell colony that you could then grow. There is a problem with this because cells can revert and become streptomycin resistant. So if you were trying to figure out how many cells in this solution were streptomycin resistant initially, you wouldn't really be able to tell that because you might have 3 cells in here that are streptomycin resistant, and you might get 4 colonies that will all look the same as that initial colony. But, I mean, all the cells on here are now streptomycin resistant, so it's still direct selection. The other is called indirect selection, and so we use this when we're trying to identify auxotrophic cells. So instead of finding a cell that has some type of resistance, now we're trying to find a cell that has the inability to grow without certain benefits. And so the test or the steps we use for this is known as replica plating. And so here you see a media that contains complex nutrients so all organisms can grow. So on here you're going to have the prototrophs, which are those cells that don't need any special nutrients to grow. They grow effectively in pretty much everything. And you'll have your auxotrophs, which is what you're looking for, cells that have the inability to grow without specific nutrients, okay? So once you have all the cells growing, so you have lots of colonies here, you then take a sterile velvet cloth and you blot that cloth onto the agar. Now, you have to make sure that you keep the cloth in the same position. So like we've marked this is the front of the colony plate. We have this marked here, so we know this is also the front of that colony plate, right? We then take that cloth and we put it onto a sterile nutrient agar with the same mark here, and a sterile agar that doesn't contain certain nutrients. So this is a glucose salt agar. It doesn't have all the nutrients required for growth of certain bacteria, certain auxotrophic bacteria, I should say. We plate them in the same spot. Then we grow them in an incubator, and we look for areas in the nutrient agar where we have colonies that we don't have colonies on the glucose salt agar. So in this case right here is an area where there's no growth, but here you see that there's a colony. So that's the colony you would pick up because it's considered an auxotroph. Once you get it, you then can grow it on its own, and now you can run different tests with it. And so why do we want these auxotrophs, Why do we care about having bacteria that can't use certain nutrients? That makes no sense, right? Well, we do it because if we have auxotrophic cells, we then can screen for chemicals, new chemicals specifically, that could potentially be carcinogenic. So anytime new chemicals are coming out on the market, chemical, it could be chemical like makeup, it could be body wash, it could be a chemical that sanitizes dishes, or that is used in gardening. It could be any, for any type of purpose. We want to make sure that that chemical isn't a serious mutagen, because if it is a serious mutagen, then it could also be a carcinogen. And so this is where we use a test called the Ames test. But using an Ames test, we have to first get auxotrophic cells, because the only way to run an Ames test is using specific auxotrophic cells. And so one of the most common auxotrophs that we use is histidine requiring Salmonella Auxotrophs, so histidine dependent Salmonella species, okay? And we grab the bacteria that we want to use and we put them on a plate with the chemical, and if they grow without histidine, that's most likely considered a mutagen. So let's look at this. So here we have a control plate, and here we have the test plate, okay? Control, test. Both of these plates are histidine requiring plates, so they don't have histidine, all right? We add bacteria that are histidine dependent, which means they won't be able to grow on these plates. We then, to the control, add a liquid that does not have the mutagen, so probably something like water, right? But over here, we add a liquid that has the mutagen, so maybe water plus mutagen, okay? So this is whatever chemical we're testing. We then incubate the plates. So if we do, so there's always that chance for spontaneous mutations occurring. So here we have, that control plate has 2 colonies of histidine, of Salmonella that can produce their own histidine. So if we have very few colonies growing, so there's 2 over here, there might be 3 colonies over here, that is going to be it's going to tell us that this chemical is likely not a mutagen. Whereas if a lot of colonies are growing, so here you have lots of histidine reverence, or revertance, what that means is some type of mutations have occurred. So this one is indicating that whatever chemical you use, whatever this mutagen is very likely also carcinogenic. It doesn't say it is for sure carcinogenic, but the more mutagenic it is, the more likely it is also carcinogenic. All right, I'm going to stop here, and we'll get into how we can transfer genes from one organism to another through 3 different processes in our next video. Bye.